I hope that you hear Dr. Calendar's cases and realize that this isn't just happening here. Myeloma physicians consult with each other all the time, particularly for difficult cases. We may not always be in the same room together, but I think this is really the beauty of being able to collaborate. But also shout out to clinical trial participation, particularly because hopefully this allows you to have access to therapy sooner, but also allow us to continue to gather information and really be better at what we're trying to answer the questions in the future.
If you notice, the title of our meetings are round tables, 2 words, not round table. And this is the round table portion. And the reason for that, I want you to envision the King Arthur's round table where everybody's got a front seat. And this is where you are all equals today on this section. So I'd like for you to think about any questions you may have from this morning. We'll do this like an auctioneer thing. You just give me a little signal and I will let you know who's up next. And we will go for the next half an hour on this. So who would like to be the first? Oh, it's always the first one is the hardest, you know. I'm going to go here and here. Okay. I'll hold the mic so that we don't pass too many germs.
You can hit me with it. I have a facetious question. Why is it when I have a loud voice?
But we need it for the recording.
Why is it when you ask a doctor or a myeloma specialist what they would do in a particular situation, if you ask 10 myeloma specialists, you get 12 answers? I'm serious.
Well, I'm going to, obviously that's a little bit of a throwing down the gauntlet, as they say. I mean, I think my experience is it depends on the situation. Obviously, when we were just talking about that previous case, I would agree with you. There are some strong feelings about these precursor myeloma stages, whether they should be treated or not. Most of the time, we will agree that if somebody has symptomatic myeloma, they need to be treated. But what they won't agree on necessarily is what that initial treatment should be. And you will get a lot of opinions about that.
Yeah, I think that when we have we have a tumor board every Friday that has about 18 doctors on it. And I'd say that 90% of the time we're pretty much in line with each other. That I think there is very strong data for a lot of things with myeloma. And just like everyone can interpret it a bit differently, but mainly I think that we're in cohesion. It gets a little bit muddier when you deal with more rare things where the data isn't as robust. So, smoldering myeloma, again, is a perfect example of that. I looked, and of all the myeloma papers that have been published, only 2% have, and this is over history, this is over the past 40 years, have smoldering in the title, which means that there's a paucity of information. And when there's a paucity of information, you're going to have different views. When you have, first relapse, lots of data there. When you have newly diagnosed, lots of data there. And then the opinions become more congruent.
What about the difference between yes for a stem cell transplant or no for a stem cell transplant?
Every time that we get new drugs, we keep going back to ask ourselves, can we just let go of this transplant already? I sure wish that there's a way to not ever look at high-dose melphalan and toxicity again, but so far what we have learned over and over again is with myeloma, using different tools to combine together to have that eventual goal of reducing the cancer cells to the bare minimum and then keep them there. Continues to benefit from having transplant kind of included on that treatment choices. So do I see us not doing transplant? Could be in some population. Do I see it not ever used again? That may take some time to hopefully move our very, very new therapy right now to earlier line of therapy and prove that they surpass the outcome that transplant, which has been, by the way, around for a few decades, it has to actually prove that we no longer need it. So stay put. But I understand transplant's not for everyone, right? So for people whose health don't allow that to happen, patients whose social circumstance is not allowed for that to occur. For right now, we have some glimpse of early data that maybe patients who have very, very, very good remission, those folks who Dr. Callender was talking about, this newer technology, this MRD negativity, if they were already there after they're getting their so-called combination 3-4 drugs that Dr. Lee was talking about, maybe they were the group that were already projected to do very well and maybe collecting their stem cells and hold back on transplant and be under good close observation and bring on the transplant if their cancer would try to misbehave could be an acceptable choice. But just stay tuned. Time will tell.
I think the really exciting thing, part of the reason that there may not be congruency between opinions is that this is evolving. Myeloma is evolving so quickly. When I talk to my colleagues about other tumors, there's, I think for like pancreatic cancer, they pretty much have the same idea. All the doctors have the same idea for that cancer because there's not a lot of positive data. It doesn't evolve very quickly. And so they're very homogeneous in their opinion because I hate to say that, but nothing's happening in their disease. Myeloma is just ongoing. I mean, it doesn't quit. And when it evolves that quickly, then it's hard for different people to keep up with different areas, and you can get a difference of opinion. But that's a good problem to have it evolving so quickly.
I would just say, to touch upon some very important issues that Dr. Cole brought up earlier, there's actually 2 very important studies that are out there looking at transplant in African Americans. And one is looking from, and you may have been involved in this, looking at differences in outcome of white versus Black patients. And in an analysis by the Multiple Myeloma Research Consortium, one of the things that stood out is not getting a transplant. And that was one of the big things that actually affected survival of Black patients. But the second thing is, if you look at a study that came out from the VA, and I actually am somebody who works at the VA for part-time, the VA has no barriers, obviously within their system, but outcomes for white and Black patients are identical. In fact, they're better if you're Black, alluding to some of the things that Dr. Cole mentioned about biology. So I do think of transplant as a very good and important thing, and I'm a big backer. It's also hilariously now one of the cheapest myeloma treatments that we have because it's got a beginning and an end. And so there are countries around the world who really embrace transplant because it's less expensive for them than all of our new drugs.
Dr. Lee, do you have any comments?
Well, I would just, I had thought,
Is it working?
I don't know, is it working?
Yeah, now it's working.
You can hear me breathing. I would just remind everybody that multiple myeloma, the multiple part is in fact real. And so that I think that the thing that we try to convey is that your myeloma is not necessarily the same as somebody else's myeloma. So, and the idea that it is, even within the same patient, it's different. Again, the idea that there are multiple clones, there's clone heterogeneity. So I think that a lot of what precision genetics and genomics have allowed us to do is actually begin to subset. It's not I'm being blinded. It's not a monolithic disease. And I think that there are going to be lots of different subsets, and that means that we need to be able to tailor somebody's therapy to their myeloma. So you will get different opinions, even in the same facility, seeing the same doctor. It may be that, well, gosh, the best treatment for your myeloma is X. Same doctor may say to another patient, the best treatment for your myeloma is Y, based on what we understand about your myeloma being different than somebody else's.
And if I could just add to that, Sal, I think the lack of consensus is a sign of incredible progress because, one of the jokes I always say is if I could transport the person I am today back to 1997, I could have treated myeloma.
Yeah.
Because it was easy. We got this thing called transplant. We got this new drug called Iridia, and there's this crazy guy in Arkansas who's using this thing called thalidomide. Let's give it a shot.
Yeah.
That was the treatment. I mean, that's facetious, but that was pretty much the treatment of myeloma.
Yeah, 1970, it would be, hey, here's some melphalan and prednisone. Be on your way,
And that's simplicity, and if that's the kind of simplicity you want, I don't want it. Next question.
Oh, okay, sorry. So I have a couple of questions. Both Dr. Cole and Dr. Savannah Sanka agreed not to treat the smoldering patient that Dr. Callender described. My question is, would a maintenance approach be advantageous in that patient, like putting them on a maintenance drug like Revlimid? I've heard that out in the field. I'm just questioning that, asking. And then I'm curious as what would a baseline MRD test look like in that smoldering patient?
Well, no, number one, I know that Dr. Cole said there's only 2% of papers out there with smoldering in it. And that's probably because, like Dr. Sanka said, it's a very rare entity. But there are 2 randomized studies, both of which used lenalidomide, one of which used dexamethasone, that showed a survival advantage. Well, actually, I shouldn't say that. Progression-free survival, one showed a survival advantage, and the one in the U.S., which is a protocol that Dr. Loniel led from Emory, does give lenalidomide indefinitely. And although in reality, the average time people were on treatment was right around a little bit less than 2 years. But so there is a precedent for that. And we have patients still on that trial who've been on lenalidomide for going on 6 years. So we'll see. But the skeptics abound is all I can say.
So I'm in the middle. They will not duke it out.
So,
But I think ultimate thing though is why are we so passionate about smoldering myeloma is because we know that once patients get symptomatic multiple myeloma, currently we're telling patients that our biggest goal is to achieve long-term remission. Hopefully they're cured if they don't have their cancer coming back and unfortunately pass on because of something else. But perhaps smoldering is the time, right, when people's body system is fit, when they don't really have end organ damage, when their immune system is more intact because they're not really having full-blown progression. Could that be a time that we can aim for a cure? And that's why this sort of duking it out happens, because really, if you wait too long, you might lose that window of opportunity. But then yet, some of these data that are out there is looking at an endpoint, something called progression-free survival, which means that if we use this medicine, we think we can delay the onset of myeloma. It's not yet proven to be prolonging their lives. And time will tell whether by prolonging the period of that remission, does that end up offering them a cure? What if that remission is so long that they don't end up with the cancer ever emerging? There's certainly a concern that this group of patients are absolutely asymptomatic. Yesterday, they felt well. Today, they're told that they may need treatment. So there is a concern about side effects, which could be cumulative if you're using the drug forever. Or at least they would have to commit to be on treatment for a while. The so-called financial barriers also there, because of course,
$40,000 a month. $40,000 a month for
So,
the rest of your life.
this sort of isn't necessarily something that's easily reimbursed and paid for by insurance company. And because I strongly believe that we could only learn whether we're doing the right thing by doing this in a more organized way. So Dr. Callender is one of the lead, she leads this nationwide clinical trial that evaluate the right approach to smoldering multiple myeloma. So just know that the only reason why they're actually having not yet similar input is because the data is coming, right? But if you and your doctor decide to not treat, just know that this doesn't mean to never ever look at the plasma cell problem. It is a continuum, it could progress, and you don't want to miss a chance of getting treated if there's a progression there. The name of the trial.
It's called the DETER trial.
But I would actually take a step back. And the questions for smoldering and the question for MGUS, in my mind, is not whether or not you should treat them or not, which is an important question. It's why do people progress? Because the whole idea of intercepting cancer progression, because we have a very well-defined pre-malignant syndrome, right? If you go all the way back, VA studies that are almost everybody that has, if not everybody, that has myeloma had MGUS to begin with, right? So if you can actually prevent MGUS, and we know that most people with MGUS don't progress, and we know that a good chunk of people with smoldering don't progress within a time frame. The question is why? And if we knew why they didn't progress, could it be the immune system? Then can we vaccinate against progression and say, all right, not only will you get, can you get prevented from actually getting a cancer as opposed to treating it early? And so I think that the biology and the research and a lot of the clinical studies are going to begin to get at the question, can we intercept cancer progression? And MGUS to smoldering to multiple myeloma is one of the classic models of disease progression. And is it the myeloma cell that's changing? Maybe, maybe not. Is it the immune system that's changing? Maybe, maybe not. And can we intervene at those levels?
Just an advertisement on September the 16th of this year, we're doing a roundtable in Boston, and the theme of the first session will be everything you wanted to know about progression, but we're afraid to ask.
We're not afraid to ask.
But I would agree. I think that that's such a good point. That in a lifetime, that 25% of smoldering patients won't turn into myeloma. And so is it into what we have talked about earlier, to give Revlimid that has a financial toxicity. As a toxicity, a lot of patients on those Revlimid trials stop the Revlimid because of toxicity. And when you look at some of the big, some of the curative trial, where I think it's like DARA KRD for smoldering myeloma, the ASCEND trial, I'm sorry, they had patients that progressed while they were on everything. There were patients still progress from smoldering to myeloma on everything, which tells us we don't know enough about the biology in applying myeloma drugs to a disease that we don't have a good understanding for. I'm a bit worried about. That wasn't too much of a fight. That was no drama.
I mean, well, to your point, there's even a trial from Spain that did a transplant. And that trial, the interesting thing to Dr. Lee's point is, if you look at the current survival in lenalidomide by itself, in ASCENT and in Cesar Gem, they're almost identical, even though one has a transplant, one has 3 drugs, and one has 1. And so, to your point, I think there's a lot we don't understand. And the other thing about the Cesar Gem studies, there were some deaths related to the treatment.
Yes.
I've had the privilege to work with Dr. Savannah Sanka for the last 10 years. I'm her nurse. And the disturbing thing that I've seen as you guys have kept people alive longer with all these new drugs is the development of plasmacytomas, the extramedullary disease in these patients. And a lot of these patients who have relapsed died without monoclonal protein in their blood, but the extramedullary disease. And so I think the criteria for most of the clinical trials is they don't have any plasmacytomas. Is that right? Or are there studies when patients relapse with extramedullary disease to see the best way to treat them?
And if you could explain what extramedullary disease is too, as well. Thanks.
Well, just one comment.
Plasmacytoma.
So the extramedullary disease is myeloma that can grow outside of the bone marrow, and that actually, to us, from a biological standpoint, actually says that the biology of the myeloma has changed, because myeloma, for the vast majority of the disease course, lives in the bone marrow, and as alluded to earlier, it lives in the bone marrow because it's got friends in the bone marrow. And one of the things that lab folks know is that if you do a bone marrow biopsy on a myeloma patient, a regular myeloma patient, and you purify their myeloma cells and you put them in a flask, they're dead in 24 hours. They're not cooking along, being happy and everything. They're dead. And that's why it's difficult to work with. The reason that they're dead is they require a neighborhood to live in. So we call it the niche within the bone marrow, but there are lots of normal cells in that bone marrow that keep that myeloma alive. And actually, we think that a bunch of our therapy actually targets the normal cells and not the myeloma. And I suspect that that's why melphalan works as well as it does. Kills the myeloma, kills the microenvironment, the niche that the myelomas are growing in. The challenge for extramedullary disease that those myelomas have figured out, I don't need to live here anymore. I can get in my RV and just go all over the place and go. And all of a sudden, those myelomas are actually selected for their ability to not be dependent on their neighborhood. That's why they show up late. That's why they are harbingers of bad outcomes, because that's the most resistant myeloma. We always say once they get out of the bone marrow, we are in big trouble. Now, myelomas can start as plasmacytomas because they just sort of overgrown their neighborhood and they're now, out in the suburbs. But those myelomas are sensitive. It's the later myelomas that pop up. They stop making antibody because you say, all right, why is a myeloma cell making an M protein? What's it doing for the myeloma? Nothing. It's actually, a myeloma cell's making thousands of antibody molecules a second for no good reason, simply because it was actually part of its biology. Why doesn't it take all that energy, turn it off, and use it to keep itself alive? Well, it's structured, it's in its biology, it can't do that. But the myelomas that figure out how to do that now don't have to waste all that energy making a protein that's no use to them. They can use that energy to grow faster, things like that. So the extramedullary plasmacytomas really are a marker of bad biology that has developed because these myelomas have been, as they say, what doesn't kill you makes you stronger. And that's true for myeloma. What doesn't kill the myeloma makes the myeloma stronger. It's figuring out how to not be dependent on the things that it needs to stay alive.
I just want to make one comment. One of the things that really bothers me being involved in trials for a long time is most trials for relapsed myeloma will not exclude you for a plasmacytoma. But what really gets in the way is sort of that phenomenon I was showing about that one patient, is that a lot of times when people are relapsing with even symptoms, they have very low levels of protein, and many of the trials ask for what appears to be like a Mount Everest of protein. And you don't want to wait for the patient to hit that target that they're mandating, because by the time that would happen, they'd be very, very ill. And so that is a real thing, when you're talking about Dr. Gormley, a very powerful individual at the FDA. So we've been arguing for years that the trial eligibility really needs to be altered, and so that patients who clearly have reasons to be treated but might not meet that protein requirement can get onto these trials.
Yeah, I couldn't agree more. I mean, I also think that as Dr. Lee was talking about sort of like the different tribes, the tricky business about this extramedullary disease is you could have different tribes in different places. So it's not as if all of these lesions, if they were to happen outside the bone marrow, if you would just have a successful treatment, they would all respond in the same way. They were almost like entirely different tribes, which also throw us off in the way of measuring response. Clinical trials are just trying to find simple ways to evaluate whether something works or not. And this plasmacytoma population really makes it difficult, right? So if you have a tumor that grows at one place but shrink in another place and another new dot in another place, is that a success or a failure? Left to be seen. So I think there's just more that we would need to understand. What are the mix in all of these different places? So you see that some of these clinical trials will try to also ask for additional sampling of these lesions because we need to understand that. Also, as it turned out, this glob of tumors that are in three-dimensional outside of the bone marrow are not very accessible to some of the newer treatments we have, particularly immune therapy, right? Because you imagine the immune cells are going to have to figure out and find a way to get in there into the gritty, nitty center of the tumor mass, which then gives us sort of worse data in this group of patients because we would need to see them shrink to be able to say that these tumors are responding. While if those folks who don't have this plasmacytoma, we will use their blood tests, so much simpler to determine response. A variety of reasons just make us needing to do a whole lot more in this group of patients for sure.
Yeah, I mean, I think that some of the new clinical trials that are coming out right now really are, the new catchphrase at the FDA, and I think among clinical trialists is a subset analysis and looking at those subsets and enriching for those subsets. The trick is to reach out to make sure we include those patients on our clinical trials, because there are clinical trials looking at, oligosecretory myeloma where you don't make enough protein, and on plasmacytomas, because we need to understand that biology, because that is a different animal than the myeloma that's dependent upon the bone marrow stromal cells.
I had a patient, and I'm sure everybody on the stage had a patient. She had both bone marrow disease and extramedullary. She had subcutaneous plasmacytomas. And we treated her, and we did her bone marrow biopsy, and she had no myeloma in her bone marrow. We actually did it multiple times, but her plasmacytomas were still there, still lit up on PET, biopsy positive, still had plasmacytomas there. So she was cured in her bone marrow, we think she was cured, but she still had these plasmacytomas. So we said, what are we going to do? We just surgically removed them. So we operated on her, and she just sent me this giant box of candy. She's a chocolatier, which is why the other great thing about having your patients, she sent us a giant box of candy like last week. She's 8 years out, and so we just surgically resected the rest of her myeloma that wasn't responding to her chemotherapy. The stuff in the bone was gone, bone marrow was gone. It was the stuff that was outside the bone marrow that we just took a different approach. We went old school and just had the surgeon take it out. So, yes, that doesn't happen very often, but it certainly happened in her case. So.
Dr. Lee, did you bring enough chocolate for the rest of the class?
No, it's all gone. She's a great chocolatier. These things disappear. That box disappears in like a matter of minutes.
And one question.
Well, I have two questions, but so is there a group effort to talk about this clinical trial redesign, or working with the FDA to make these measurements more inclusive? Or that's my first question. And then my second question is, what can patients learn in order to become better patients for you?
Towards the first question, there was actually a big meeting in October in Washington, D.C. between the International Myeloma Society, that they set it up with the Food and Drug Administration to basically sort of talk about all of this stuff. And I think that there were also industry partners there too. And I think everyone agreed in principle there are a lot of things that we should fix, like thousands of labs that everybody has to do on a trial. I don't know if anybody's on a trial, but you may notice that your neighbor is probably not getting blood drawn nearly as often. Trying to get trials out to people who live in rural communities, which is a big deal in Wisconsin, Michigan for sure, here in Indiana as well. But it seems to be, in my opinion, a little bit glacial. I mean, this is not a new topic. This has been around for a number of years. And so I don't know that I saw personally a lot of action items coming out where somebody's saying, oh, we're going to do this trial now where we're just going to, if you have a radiographic evidence of relapse, that's all it is going to take to get into the trial. So I do think it's going to take all of our efforts just pounding and saying, we really, really, really need to do this. But I haven't seen any actual protocol come out yet that captures that.
Yeah, when I actually rewrote mine, when I was like, oh my goodness, don't be an idiot and cut and paste. It didn't fall. When I gave it to industry, they didn't like the changes. They wanted to stick with the old school kind of criteria. And so now we're just kind of going back and forth. But I think that that's where the future is, because, one thing that's at a premium are patients going on clinical trials. And if you continue to set the same silos and the same way we've been doing things, you're going to exclude more and more patients. And with patients being at a premium, that everybody now, thank goodness we've gotten a word out where people want to be involved in clinical trials. If we make it so restrictive, we're going to start losing patients. So there's a strong effort, I think, on everyone's part to try and make them more inclusive, especially as people live longer with myeloma and it begins to evolve differently where the biology changes. If you leave it and say you need an M protein greater than 0.2 or 1.5, let's say, to go into clinical trial, you're going to miss a lot of people in the future. So, I mean, there are a lot of both internal and external forces that are going to change, are going to really force our hand to make our clinical trials more inclusive.
Well, I think that there's another aspect. We all sit in this room and assume that the rest of the world gets the same myeloma treatment that we all have access to, right? And there are actually parts of Europe that don't get the same myeloma treatment that the U.S. gets, and certainly most of the rest of the world, low and middle income countries, don't get that treatment. But there is a very thoughtful approach going on in global oncology as to how do you do clinical trials in low and middle income countries. You can't have 7,000 blood tests drawn in Moi, Kenya, because that simply can't do that. So how do you simplify a clinical trial to give you important information on the population that you're studying? And then how do you actually develop drugs that you can actually deploy? I mean, we're never going to get CAR T cells, in Cuba simply because, geez, they don't have the technology to do that. So how do you develop new agents that can be deployed out globally? Because yes, I think it's incredibly important that we treat people where we live, but it's also incredibly important to think about how do we treat everybody else that doesn't have access to cutting edge technology and expensive drugs and therapies. How do we treat those patients also?
Well said. Perfect.
So I'm gonna
Guy with the bow tie.
Exactly. The bow tie component's over here.
So I'm gonna actually lean on that and say that the global collaboration happens. You may not think about it all the time, but as we're trying to make treatment accessible to folks across the globe, we're also learning about how biology of myeloma itself is different and how we as people react to drugs differently. So Dr. Cole was already telling you about, what we learned about the different genetic makeup of the cancer cells in different ethnicity, but also really how we respond to drug would matter as well. So we actually have the IU Kenyan program where we're pretty fortunate to have this collaboration that has been around for over 20 years and is now has expanded outside of just infection focus when it started in the HIV era to now incorporate cancer care and cancer research. So there's a lot going on. Now, how to be a patient that helped me? So there are many ways, right? But you have to know one thing. You are really, truly the captain of the ship. And the rest of your doctors are your crews. Everyone is different, right? So the goal of the treatment should be clearly established. We're not here to drive down the number of the M protein. We're not here we're hopefully here to help you be the best you can. And I think if I would say anything, it is to let us help you by sharing your concern. Tell us what your goals are, what are the barriers of your treatments. If you need to work every day, then your treatment should happen at the end of the day. Or, if we're worried about the steroid keeping you up at night and your spouse locked you in the room because you keep pacing the house, I don't see that, so you're gonna have to tell me, right? So we're truly the partners in this, but also in the 15, 20 minutes of time that you have with your doctors, it would really help to have the sort of things that we need to tackle, things that are worrisome for you, things that you wanted to address so that we can really make use of that face-to-face visit. That's what I've learned to be useful.
Yeah, I think the, how not to be a good patient is when you say, Doc, I'll do whatever you say. Because it's a partnership. I mean, I work really hard to establish partnerships with my patients, and that we negotiate, we discuss, and we discuss options and come to a consensus between the patient and I about what our next steps are. And so, because I don't live in your shoes and I don't know what it's like to go home, how important it is to walk your dog, how important it is for this and that. And if you leave me to make that decision, I'm dealing with a very small amount of information. Even if I've known you for years, I don't know what it's like when you go home at night and what it's like. The best person that tells me what's going on with you is you. And so we come to a partnership. And so just to say, do whatever you want, really worries me. I mean, that raises a lot of red flags. The other thing I love is when patients write down questions. I usually, when I prepare, when I go into a room, I kind of have a list of the questions and what we're going to, the goals of this visit. When patients have that, then we hit those things very quickly. Then we can talk about fun things like chocolate and things like that. So I think having a list, but also advocating for yourself and knowing and getting that partnership established between you and your doctor is so rewarding for me, because I get to share good news. I mean, half of the visits that I have, they're like, tell me what's new, Craig. And I said, well, here we go. Here's what's new. And I'm excited to talk, because my wife won't let me talk about myeloma anymore. She's like, done. So who can I talk to? I talk to my patients, and that is it's good for everyone to hear all the good news that's happening.
Just the last thing I would like to say towards that, please don't be shy about telling your providers about bills, because sometimes we are oblivious to what is getting paid for and what not. So not too long ago, a patient came in to me and said, what is this fish test? And I said, well, explained as we talked about it. And they said, well, my insurance refused to pay the $4,000 bill that came in from pathology for this. And they were just going to pay it. They were just going to, get on. And, obviously we were able to go in and say, this is standard of care. This is something you need to cover. But I think it's really important for your providers to know if you are really getting stuck with something that's huge, because often we don't know that.
And I would say, and I'm preaching to the choir, the more you know, the better. And I utilized the HealthTree University thing to learn about stuff, because you can imagine you've been sitting here all morning. We've been blabbering on about next generation sequencing. I hope it's beginning to settle in. If your doctor wanders in for 5 minutes and says, hey, we're going to do a next generation sequencing test, it'll be great, and then walks out, or he or she walks out, and you don't know what that means, you're at a huge disadvantage. And there are great resources out there through HealthTree, through to learn about these things. So you want to go in prepared, and as everybody has alluded to, the field is moving so quickly. When I was a medical student, right, before they invented electricity and penicillin, right, that it used to take, somebody said, the time it takes from a phase 1 study to getting it FDA approved was 10 years, right? I think what the BCMA CAR T cells were approved in 18 months, 24 months, right? They started at a phase 1. Less than 2 years later, FDA approved them. If you didn't know what that was about, you missed out on one of the biggest recent advances in myeloma. So it's important to keep up to date. It's important to know these things. The amount of technology, the approaches, both diagnostically and therapeutically, are accelerating. We think that's great, but I think that it puts patients at a disadvantage, especially if they haven't kept up and they've got 20 minutes, 10 minutes with their doc, and their doc's not going to sit there and explain, all right, this is how we stratify your disease based on these mutations we found, right? So I think that the more you know, the better.
Well, thank you. I hope you all got something out of this morning, and we're going to get a lot more out of this afternoon. One of the points of this program is that it's not important for you to be a medical expert, and I hope you don't feel you have to be. But it's really important for you to know how to ask, to know what you don't know, to know how to ask questions about them, and that will help your treatment tremendously.